Hello, this is Adam Pawson welcoming you to this bite-sized bio webinar, which today is sponsored by Molecular Machines and Industries. Molecular Machines and Industries is a leading provider of microscopic single-cell isolation tools. They specialize in single-cell separation, laser microdissection, and optical tweezers for a wide range of clinical and research-based applications in life sciences, material sciences, and healthcare. Today's presentation is titled Visualizing Plant Viral Infections at Single-Cell Resolution and is being presented by Dr. Bjorn Krenz and Christoph Sicking from the Leibniz Institute DSMZ-German Collection of Microorganisms and Cell Cultures GmbH. Dr. Bjorn Krenz is a senior research scientist at the Leibniz Institute DSMZ specializing in plant viruses. He has an extensive research background from the University of Stuttgart to Cornell University, USA. He previously led the Virus Interact group at the DSMZ and has contributed to key publications in virology. Christoph Sicking obtained his master's degree in horticulture at the University of Hanover with a focus on plant virology. Since 2020, Christoph has been working on his PhD at the DSMZ. As always, we will have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears on the top right panel of your screen, and I'll put them to Bjorn and Christoph at the end. Details on how to access the on-demand recording of this webinar will be sent to you by email shortly. So now over to you, Bjorn and Christoph, for the presentation.
So thank you, Adam, for this kind introduction, and hello, everyone. Welcome to our talk, Visualizing Plant Viral Infections at Single-Cell Resolution. So my name is Bjorn Krenz, and with me today is Christoph Sicking, my PhD student at our department. Christoph and I are coming to you from the DSMZ. That's one of the world's premier biological resource center located in the city of Braunschweig in Germany. Our mission at the DSMZ is quite fascinating. So we focus on collecting and providing a variety of biological materials, like from bacteria to plant viruses, and also supporting research with our expertise. So Christoph and me, we work for the plant virus department. We are proud to house an extensive collection of plant viruses and offer state-of-the-art diagnostic tools, like accredited ELISA tests, for example, or kits, or we perform virus discovery by next-generation sequencing or high-throughput sequencing. So our work is also at the forefront of virus research, from gene-level studies to understanding plant responses and diseases. So today we're going to walk you through the realm of plant virology, starting with begomoviruses. We'll look into how we use laser dissection microscopy to go from looking at whole tissue to zooming into tiny organelles, like a nucleus. We will explore the practical applications of the laser dissection microscope in analyzing nuclei isolated from virus-infected plant cells, which offer us insight into viral infection mechanisms. And lastly, we wrap up with a summary of our key points and open the floor for any questions you might have. So plant viruses are important components of the natural ecosystem, and also some might not affect plants. Some are causing diseases in crops and therefore present serious threats to food and feed, and thus impact the livelihood of many people. With economic implications in the billions, understanding these viruses is more crucial than ever, especially the ones spread by whiteflies, like the begomoviruses. So these are the bad guys we are up against in the battle for our crops. Begomoviruses are responsible for a range of plant diseases, in particular in tropical and subtropical regions, affecting crops like tomato, beans, and cotton, with symptoms including yellowing, stunting, and leaf curling. Begomoviruses are belonging to the plant virus family of Geminiviridae. They're characterized by their twin-shaped particle structure, which harbors a DNA genome. More precisely, they possess single-stranded DNA. Which is circular in structure. They can be either monopartite with a single genomic component or bipartite with two components. So we focus on the study of the mosaic virus. It's a model virus for bipartite begomoviruses for us. And we want to understand how it interacts with the plant host and how viral diseases do their thing at the molecular level. In particular, we address the question, how does ABMB manage to establish itself in a host plant? And we want to investigate the very first steps that lead to a successful infection. Therefore, our approach is first to identify infected cells, which involves an attenuated ABMV, where the code protein, you can see here, where the code protein is replaced by the open reading frame of the green fluorescent protein. So this modification allows us to visually track the infection in plants by GFP fluorescence, providing a direct method to monitor and study the infection dynamics at the cellular level. Since ABMV is flame limited, we utilize the so-called clear signal technology to basically make the tissue transparent for visualization. But to work with these cells, and not only to monitor them, we require specialized techniques that can isolate pure samples from the complex mixture of cell type within a plant tissue. This isolation is crucial for a biologically meaningful analysis of the infection process. And to address this, our group utilizes this solution here, the cutting edge solution, in my opinion, the MMI cell cut and selector system. It's a combination of two systems you can see here on the picture. So this system allows us for precise laser dissection of specific cells under the microscope, enabling us to extract and collect pure samples of infected cells from the cellular mosaic of plant tissue. Besides, the laser dissection microscope techniques we employ is versatile and extend beyond the study of virus-infected cells. For instances, we use it to propagate oscillatoria, an unusual approach, I would say, or we use it to work with a fungal pathogen responsible for a potato disease, which you can see here. It's a potato wart disease. So these applications show the broad utility of the laser dissection microscope in various areas of the plant biological research. In addition to the MMI cell cut system, we have it combined with the, as I said, with the MMI selector system, which allows us, for example here, also the isolation of protoplast. For example here, it's a potato protoplast, which were collected individually, and to analyze the genome, for example, which was edited by the CRISPR technology. You can see where we have picked up one protoplast, and then we release it in the collection a while. But coming back to the cell cut system, here we must keep in mind, so we must envision the samples as quasi two-dimensional. So we need to prepare tissue sections, and we use a cryotome for this. The optimal laser dissection results we obtained when working with tissue sections that are sliced to approximately 50 micrometers in thickness. And this specific sectioning allows us to target and isolate individual cells, like depicted here. In this case, we isolated phloem cells from Arabidopsis thaliana petioles under fluorescence. And here to analyze, in this particular case, the expression level of the G3BP protein family, which we have published here, 2021. So in principle, what you see is what you get. When we are looking at tissue samples at the molecular level, we sometimes run into the problems caused by fixatives like formalin. So these chemicals can create, or these chemicals create, for sure create, crosslinks that make it tough to get or to copy DNA or RNA. Science is always moving forward, and now we've got a new way to pull it out and amplify genetic material from this tissue. Nevertheless, so actually Christoph Sicking, our PhD student, now wants to present you a method which has been developed to avoid fixation. And that's why we now switch to Christoph.
Hello, everyone. Also from my side, I'm happy to present you some of my results I've produced with the MMI cell cut system. So here our lab has developed a method to work with actually living cells. So this approach therefore avoids the issues associated with fixation and allows us for studies of cellular processes as they occur. Here, for example, we infiltrate Nicotiana bentamiana leaves with agrobacteria, which are harboring a GFP-marked ABMV clone, as Björn recently mentioned. And this leads to a GFP expression of the infected cells. We then can peel off the epidermis, as you can see in panel B, place it on the special slides for the laser dissection microscope, and then we can cut it with the cell cut system and collect the material of interest in the sample tubes with these sticking lids. Additionally, we can really mark precisely these GFP expressing cells, as you can see here in the video. Besides, it's a little bit speeded up. We can very precisely mark the shape of the cell and cut it really precisely, that we only have the cell that we wanted to in the tissue, in the tube. And as you can see, it's very convenient and easy to handle and easy to mark our region of interest. In the next step, as you can see here, we are going to dissect the cell. Of course, this is another example as shown previously. So the cell is another than previously marked, but the selection process is identical. The laser cuts out exactly the cell we wanted to investigate. Sometimes, as you can see in a second, it has to cut twice. This is because the tissue is maybe thicker in one point or another, or it's just not completely in focus, but you can adjust the parameters of the cutting to ensure the successful sampling. And here you can see the cell is gone. And then after cutting and collecting, we can double check and control whether the cell is really being successfully transferred to the sticking lid, as you can see here in the last picture. Besides cutting whole cells, we can also go for single nuclei. As you can see here, the nuclei is marked out by this strong GFP signal, and the laser then cuts out only the nucleus with a round shape. Collection tube comes down, and you can see the hole in the middle of the tissue, and then we can work with the cutted out nuclei. So all in all, after double checking for transfer for either the cell or the nucleus, we can use this collected material for either RCA, RFLP, or qPCR analysis. This RCA method is a qualitative method where we can prove if there's virus present or not, and qPCR is also method where we can quantify. So how can we perform our analysis in the tubes? For both approaches, we start with releasing the sample material from the sticky lid of the collection tube, as you can see in the most left panel. Therefore, we place a little droplet of approximately 10 microliters of sample buffer directly on the lid. And in the next step, vortex the sample heads down to release the sample from the surface of the collection tube. And then we can go for either incubating the RCA reaction on room temperature, or perform the qPCR analysis directly in the thermocycler. Here you can see in panel B the representative result of our qualitative proof using the RCA, followed by a restriction digest, and the fragments which were produced using this method have been visualized on an agarose gel, and you can see the precise and specific bands for the ABMV infection in panel B. So yes, we are able to prove the viral infection upon few nuclei or a whole cell. And additionally, in panel C, you can see that we can also quantify the number of ABMV viral DNA copies, which started at few hundred copies per nuclei and go up to a few thousands. But of course, we are also interested in working with the wild-type virus. Therefore, we need a plant marker protein that shows infected cells or nuclei, similar to the cells marked by the GFP in the previous example, with the attenuated ABMV virus. We have chosen to work with the H2B protein, which belongs to the chromatin complex and shows us very nicely an alternative pattern and relocalization to the nuclear periphery compared to mock conditions. When it comes to a viral infection, either with ABMV or TYLCV, which is another begomovirus, we can see a different GFP signal than in healthy nuclei. And we use this relocalization to localize infected nuclei in peeled epidermal tissue. We can then specifically mark them, cut them out, and collect them, and perform the same analysis than we were using with the ABMV GFP, but for this case, for the wild-type viruses. So again, the RCA or qPCR analysis can be performed. I will show you in the next slide the RCA results. Here you can see that we can really prove the ABMV and TYLCV infection upon nuclei that show these H2B relocalization, and we only need few nuclei, so 3 to 5 nuclei are really enough to give us this specific band pattern that shows us that these nuclei are really infected. But we are not only interested in looking at infections on single cell level, we also want to understand whether plant DNA viruses undergo nuclear egress similar to animal circoviruses, and therefore we want to understand or investigate if they destabilize the nuclear lamina or the nuclear membrane, or if we can find any alterations in the fatty acid composition of infected nuclei or in the whole epidermis. And this work was done by our master's student, Ina Schmidt. Therefore, she used an inverse approach, so to say. So we again peeled the epidermis, used the ABMV GFP or cabbage leaf curl GFP, which shows us then which cells were viral infected with the GFP signal. We then removed with a laser the nuclei and analyzed the tissue within our metabolomics facility. And we could then really show that there are changes happening in the lipid composition of infected and non-infected cabbage leaf curl tissue, but only in some cases. So there's more work to be done, and but it's an interesting field of view. We can use our LDM techniques to either analyze tissue-specific gene expression, or we can also prove out of single cells if they are infected by a virus, and can also quantify this amount of viral DNA. And additionally, in collaboration with our metabolomics facility, we can perform comparative lipidomics. And last but not least, we want to thank our former group members, Dr. Khalid Amari, Henrik Reuper, Verena Mayberg, and Ina, for working with us and collaborating. We want to thank the whole DSMZ plant virus department, headed by Dr. Stefan Winter, and our collaboration partner for the lipidomics analysis, the working group metabolomics.
Yeah, and we thank you for listening, and we are happy to take any questions you might have.
Okay, well, thank you, Bjorn and Christoph. That was an excellent presentation, very interesting. We will go to the first question from the audience. So, Bjorn, Christoph, the first question is, what is the size range of the isolated plant nuclei, and what are the smallest possible structures that can be isolated with laser microdissection?
Okay, first of all, thank you for giving us the opportunity to share our data and show you how our laser dissection system works. The sizes of nuclei, they range between 5 and 50 micrometers. It depends a little bit on the plant. So the pictures you have seen or the videos you have seen have been done with model plants like Nicotiana bentamiana, tobacco variety, and Arabidopsis thayana, and they roughly have the epidermal nuclei have a medium of 10 micrometer. And so 10 micrometer, as you can see, or as you have seen in the videos, can be nicely isolated. The chloroplasts, for example, they're roughly 5 micrometers. You can also isolate the chloroplast. Everything which is underneath 5 micrometers is rough, so it's possible, but I would say 5 micrometers. That's fine. Yeah,
okay,
that's
great.
cool.
Thank you. Thanks a lot. Okay, and then on to the next question. To ensure consistent and reliable results in downstream analysis, what is the minimum number of individual nuclei that should be pulled together?
Oh, okay. I mean, it depends on the downstream application. Yeah. So what you have seen here is a qualitative proof if the isolated nucleus is infected or not. So this is a yes or no answer. And all you need here is a reliable amplification step, which is where the RCA works perfectly, or the PCR or qPCR. But I must say, if you isolate one nucleus, it's always. I mean, also we also double check the lid if we have something in the lid, or that we have isolated the nucleus. Sometimes the whole procedure might be like this, that you can lose your template for proper amplification. So that's the reason why we usually pull 3 nuclei. And with 3 nuclei, you always get 100% amplification. And then it really depends on your downstream application, what you have, and you have to do the statistics, and it must be significant. But it needs some time to collect those nuclei and to get reliable results. So I think in total, I mean, how many have you been isolated, roughly?
Several hundreds.
Several hundreds.
Okay. All right, brilliant. Thank you very much. Okay, so the next question, a really interesting talk. Thank you. What objective did you use for dissect, and what is the thickness of the laser cut? Were you able to?
I say it again. I'm sorry.
So, what objective did you use for the dissect, and what is the thickness of the laser cut?
Okay, so I think we used it the 20 times or the 40 times?
You can use both. It depends on the target you're looking at. So for dissecting whole cells, the 20-fold would be the best choice. But you can also go for the 10-fold. But I've mainly worked with the 20-fold. You can also use the 20-fold for dissecting the nuclei out of one cell. But yeah, if you want to investigate structures that are smaller, you would have to go to the 40-fold. But yeah, 40-fold is the biggest magnification we have in our system. The thickness of the laser is beneath.
It's about roughly 1 micrometer.
Yeah.
Sometimes less. Yeah.
Okay, thank you. So the next question, can you collect all cells that you cut? How do you solve this LMD problem?
Okay, so I mean, it depends. You can collect the cells sequentially. Yeah, so you cut out, collect, cut out, collect. You can cut several cells and then collect simultaneously. Yeah, that's possible. But I mean, we do it sequentially. Yeah, just to be on the sure side. So when the laser goes around one cell, for example, sometimes due to the different height of the cell, yeah, and sometimes some is a little bit sticky. You have to cut sometimes twice. You go around the same edge or the same borders again, and then then you can collect nearly every cell. Yes. Sometimes the sample is a little bit not very helpful. Let's put it this way, and then you just take other samples, another peel, and then it works perfectly.
All right, thanks, Bjorn. Okay, so the next question, this is quite a long one. So what do you think does GFP have any role on the localization of viral proteins? Did you quantify the viral titer in both cases, like with the nuclear marker, the viral protein without a tag, and compare it with the GFP tagged viral protein?
Oh wow! Yeah. So first of all, when we use what we call a GFP virus or an RFP virus, so the genome of the virus is altered. So in this case, with ABMV, you replace the code protein with the open reading frame of the GFP. So the virus is attenuated. Yeah, it's not the wild type virus, and then the titer is lower. Yeah. When you use the wild type virus, the titer goes up. Yeah. I don't think that the GFP itself has, or the fluorescent protein or any marker protein has an influence on the amount of viral DNA. But it would be hard to prove other, right? So what do you say?
Yeah. So it's also interesting to look at how this GFP mutant performs in comparison to the wild type virus when it comes to the amount of viral DNA for component A and B simultaneously. So this could also be an effect of the replacement of the GFP of the replacement of the cod protein with the GFP, and we are currently working on establishing a multiplex qPCR approach where we can then quantify the amount of both genomic components out of one sample, since the LMD collected nuclei or cells are just one-shot sample. So once we have analyzed them, they are done for analysis.
Yeah, the sample is burned.
Yeah, sample.
More or less.
Yeah. So it's a destructive method. So we cannot do another qPCR on the same sample for component B, for example. And therefore we are currently working on this problem that we can solve and tackle this question.
Okay. That's great. Thanks, Christoph. So the next question, can we use LMD for animal or human cells which can be infected with virus?
Yes, of course.
Okay.
Yeah. I mean, I haven't done it by myself because I'm a plant virologist. But I don't see any limitations to put an infected animal or human cell on a slide and then cut it out. Yeah, I mean you have to watch the safety instructions here, of course. Yeah, because plant viruses don't infect humans. Yeah, so here, but technically there's basically no difference.
Okay, thanks, Bjorn. Right, so the next question, what specific tool did you utilize for isolating the individual protoplasts?
So this system is called Selector from MMI. Yeah, and it's a capillary-based equipment which transports nanoliter. Yeah, in the range of nanoliter, they basically suck out, in this case, protoplasts, potato protoplasts, what you have seen in the, on the slide. And yeah, you transport it in nanoliters, more or less, so that's a very, very short range, roughly in the size of 60 to 80 micrometer. And you carefully suck it into the capillary, yeah, and then you dispose it in a collection vial. What else can we say to the system? I mean, it's not restricted to protoplasts, so it's basically to get something out of the suspension. Yeah, you can also, for example, isolate bacteria, yeah, or protists, or anything in a solution. You can also use the cell selector system to move animal or human cells. But here you have to ask the MMI guys for this. Yeah.
Okay, thanks. Right, so another one, also a bit long, so listen up. So I think this technique has a limitation. The tissues used for cutting must be almost 2D, such as the epidermis. And according to my own experience, we cut pit fields from cell walls. Have you tried any 3D tissues, and how did it work?
Yeah, that's basically what I said in the talk. We basically have to think two-dimensional here. Yeah. So with the peeling of the epidermis, it's one cell layer. It's really one cell layer. And here it's basically to the or let's put it this way. It's thin enough, yeah, that the laser can cut properly. There's a drilling function, yeah, so you can actually go deeper, yeah, so we go around the border or the edge of the cell, yeah, and then you shift the focus a little bit, and they can actually drill deeper, and use a thicker tissue or a thicker sample. But I never have tried really a 3D, yeah. So, for example, when we stick to our research, yeah, and we want to investigate a systemically infected tissue, yeah, and then the virus moves within the vascular tissue and is, for example, phloem limited, yeah, then we really would need to fix and section the samples, yeah, to reach the phloem. Yeah.
Okay, thank you.
I hope this answered the question.
All right. So the next question. Could you provide some insights into the time requirements for isolating a single cell or nucleus using laser microdissection?
Yeah. Also, so the good news is you get better and better and quicker and quicker. The drawback here is the samples are always a little bit different. Yeah. So for example, when you peel, there's it sticks to the slide or the foil, or then it also needs to dry a little bit. So here the time can vary. Yeah, but I think once you get into the system, once you establish a sort of routine and you're familiar with the handling, so I would say that collecting, for example, a cell is within minutes. Yeah.
Yeah.
Do you want to add a precise time?
So yeah, if you work with living tissue like the peeled epidermis, it's not so time consuming compared to fixing and cutting, or fixing, embedding and cutting, and then performing the collection or the dissection. We can reach one cell in minutes. Once we have the sample, it's
it's quite quick. I mean, I would say if you want, for example, like 25, 30 cells, yeah, you would need like half a day probably. Because you have to set up the system, you have to prepare the samples and everything. I mean, it's not meant for high throughput. Yeah. Here you need different approaches.
Right. Okay. So this is possibly related to that. Then, is it feasible to simultaneously quantify multiple virus infections within a single sample?
Yeah, but this is what Christoph just briefly mentioned. So that's the method of a multiplex PCR, for example, where you use different dyes, or the different dyes can distinguish between the amplification of different kind of RNA or DNA source material. Yeah, I'm not quite sure if it will work with RNA and DNA viruses simultaneously. Here depends on the sample preparation, I think, but for different kind of DNA viruses, and you have specific primers, you have the machine to visualize or to measure the different fluorescent dyes. Yeah, then it's doable, but you have to do a lot of controls.
Okay. Well, thank you, Bjorn. Christoph, I think we're done with all the questions for today. So that brings us to the end of today's webinar. Thank you for a very illuminating presentation and a great discussion. There were plenty of questions there. And thank you also to our sponsor, Molecular Machines and Industries. And finally, thanks to you, the audience, for taking the time to attend and listen in. So until next time, good luck in your research and goodbye from all of us at Molecular Machines and Industries and Bite Size Bio.
Yeah, thank you much for listening. Thank you, guys.
Bye-bye.